Good morning, good afternoon, and good evening, everyone, and thank you for joining us for our webinar titled Case Studies in Home Cage Monitoring, Rodent Behavior, Circadian Biology, and Animal Welfare. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event. Our session is sponsored by Technoplast and will feature Dr. Kenneth Dyar, Senior Scientist at the Helmholtz Diabetes Center in Munich, and Dr. Joanna Moore, Investigator at GSK, who will present applications of automated home cage activity monitoring and discuss how it can be used to improve animal welfare, optimize study design, and drive animal behavior and physiology research. And with that, I'm very pleased to introduce our first speaker, Dr. Kenneth Dyar. Ken, thanks so much for joining us today, and feel free to take it away whenever you're ready.
Thank you, Liam, for the introduction. Hello, everybody. My name is Ken. And as Liam said, I study circadian regulation of muscle metabolism here at the Helmholtz Diabetes Center in Munich. But I guess my main research focus is to understand the physiological significance of these 24-hour gene oscillations that we see in different skeletal muscles. So which genes cycle over 24 hours? Why do they cycle? And how is this cycling important for muscle health and disease? Now, using a combination of different approaches over the years, we've uncovered coordinated control of these muscle circadian genes, both by activity and feeding-dependent mechanisms. And these can act directly on gene expression, or these can act indirectly through the regulation of the muscle circadian clock. Most recently, we've begun to systematically identify all these muscle clock target genes, so the cycling genes that are directly regulated by the core clock transcription factors. And again, the ultimate goal is to understand the physiological significance of this oscillation. So how does gene oscillation link to circadian rhythms of muscle metabolism and muscle function? So just to remind you all, circadian rhythms are endogenously generated biological rhythms with a period of about 24 hours. And these include many behavioral, physiological, and metabolic rhythms like the ones you see here. So body temperature, blood flow, hormone secretion, feeding, and activity are all rhythmic processes. And over the past few decades, we've come to understand that all of these processes are controlled by circadian clocks. And these circadian clocks are just molecular oscillators, and they're present in all the cells of our body. And they're able to maintain cellular time according to the rotation of the Earth. And this is thanks to these interlocking transcriptional translation feedback loops. So in the positive loop, clock and BMAL1 genes are expressed, and their proteins accumulate and form heterodimers. And then they activate expression of the negative loop members, including cryptochromes, period, and reverb. And these negative loop members are all transcriptional repressors, so they turn off gene expression. So when these factors accumulate, they feed back and inhibit expression of the positive loop members, and binding of BMAL1 and clock to their target genes. So if you measure the RNA of these positive and negative factors across the light-dark cycle, you find that they're expressed antiphase to one another. So when one is increasing, the other one is decreasing. And this whole process, again, takes about 24 hours and is occurring every day in all the cells of your body. Now, when we talk about clock control genes, we're talking about additional target genes that are integrated into this basic feedback mechanism. And these genes can be activated or inhibited depending on how they respond to these different clock transcription factors. So for example, BMAL1, since it's a transcriptional activator, turns genes on. And reverb, since they're transcriptional repressors, are turning genes off. Gene expression profiling of different tissues over 24 hours across the light-dark cycle has revealed that the vast majority of the clock-controlled genes are tissue-specific, and these are often coding for rate-limiting steps in metabolic pathways. So while each cell has a robust 24-hour clock, in order for the tissues, the organ systems, or multicellular organisms to operate efficiently and together as a functional unit, all the individual clocks within and among different tissues must be synchronized by periodic stimuli called Zeitgebers. And this is just a German word for time giver. Now, Earth's 24-hour light-dark cycle is the main Zeitgeber for the central pacemaker in the suprachiasmatic nucleus of the hypothalamus, which is circled here red in the brain. And this light information for day and night is transmitted directly from the eye to the SCN by specialized photosensitive retinal ganglion cells in the back of the eye. And these in turn communicate the day light cycle to the SCN, which then communicates the external time to the internal clocks in peripheral tissues. And this occurs by activating the sympathetic nervous system and by regulating feeding patterns and locomotor activity and even body temperature rhythms. So in this way, the master clock in the brain coordinates the production and the distribution of a diverse array of metabolites and steroid hormones, lipids, vitamins, cofactors, and these all collectively modulate gene expression and synchronize clocks in the periphery. So this daily entrainment is needed to ensure that there's phase coherence and that this is maintained between the central and the peripheral clocks in the body. And so it's very important that we all get outside and we get a daily dose of sunlight. So hormones and metabolites are thus very important signaling molecules that are telling the clocks what time it is. And this is mostly through ligand-activated transcription factors. So the presence or the absence of these various molecules will relay the external time directly to the genome and then directly regulate metabolic gene programs. So this keeps the clocks in the individual cells synchronized with one another and in tune with what's going on with the rest of the body. So instead of all of the clocks in a tissue cycling incoherently, they're cycling together. And this really allows for different tissues and organ systems to work together as a functional unit. Of course, this means that there's a hardwired transcriptional logic and a temporal coherence that underlies normal physiology. And this has been selected for over millions of years of evolution. So living in synchrony with the environment is known to confer health and competitive advantage, whereas living out of sync is detrimental to our health and to performance. So circadian misalignment can occur when we have abrupt shifts in the environmental light-dark cycle, and this temporarily uncouples the central pacemaker from the downstream networks and desynchronizes clocks throughout the body. So a common example of this acute circadian misalignment that many of us have probably experienced is jet lag after flying to a new time zone. Now, these abrupt mismatches between internal and external time will result in sleep disturbances, fatigue, digestive problems, and impaired cognitive and physical performance. And these symptoms will normally subside after a few days of repeated exposures to new local zeitgebers like daylight, meal times, and activity or even exercise. And these all together allow for our internal clocks, again, to resynchronize to the new external time. However, modern life can also send mixed messages that interfere with this clock alignment. And this can include our work or school schedules, the medications we take, stress, whether we eat at night, or even exposure to artificial light. So watching TV or staring at a tablet or a phone at night. Now, shift workers and people who naturally go to bed later, which we call late chronotypes, are especially susceptible to these discrepancies between their biological time and social time. And this is a term that's called social jet lag. And these people often show increased risk for severe metabolic, cardiovascular, psychiatric, and neurological diseases, including obesity and diabetes. Now, studies in mice have shown that what we eat can similarly cause disease by disrupting these circadian rhythms. So if you put mice in constant darkness so that they no longer receive the external light information, you can quantify the period of their master clock in the brain by measuring their free running activity period. So this endogenous clock doesn't run at exactly 24 hours. It actually runs around 20 minutes earlier. And you can see this here in the actogram plot on the left. So when the mice are changed from their normal 12-hour light-dark cycle, written LD here at the top, and they're placed in 24 hours of constant darkness, so DD, the phase of their activity rhythm starts to shift around 20 minutes earlier each day. So their internal rhythm is maintained, it just becomes progressively out of sync with the external time. Now the mice on the left ate a standard regular chow diet, indicated by RC. But when they're switched to a high-fat diet, so HF on the right, their free-running activity period lengthens by around 12 minutes each day, showing that the period of their central clock is actually affected by the type of diet. Now, even under a normal 24-hour light-dark cycle, activity and feeding patterns are altered by high-fat diet. And you can see this if you look at the distribution of activity and feeding events over 24 hours. And I hope you can see they're no longer restricted to mostly during the dark phase. And this is indicated here by the white-black bar at the top. Now, this was especially evident when looking at the feeding patterns, which are highly disorganized, and around 30% of the meals are consumed during the light phase, when the mice are normally supposed to be sleeping and fasting. And these changes are linked to blunted circadian oscillation in the tissue clocks and altered metabolic coordination between tissues. And we uncovered this loss of metabolic coordination by measuring metabolites over 24 hours in different tissues of these mice under a regular chow diet or a high-fat diet. And you can see on the left the massive changes in tissue metabolite abundance and oscillation that are induced simply by changing the type of food that they eat. And this results in really an incoherent metabolic programming and pathophysiological metabolic rewiring between the tissues. So tissue metabolism is performing the wrong tasks at the wrong time, and tissues really aren't talking to each other anymore because their clocks are all misaligned. Now, the impact of these changes on health is obvious. So mice eating a high-fat diet have disrupted circadian alignment between the tissues, and they become insulin resistant and eventually obese. And they accumulate lipid stores throughout the body, including the liver. And this eventually causes steatosis, or fatty liver, which can lead to inflammation, liver fibrosis, liver cancer, and ultimately liver failure and early death. However, accumulating evidence has emerged suggesting that when we eat is way more important than what we eat. So in this study, already from some years ago, mice were fed a normal chow diet or a high-fat diet ad libitum, so they ate their meals whenever they chose throughout the day. Otherwise, their meals were restricted to a precise 8-hour window during the dark phase. So meals are tightly aligned with their normal physiological feeding phase. And this completely prevented the mice from eating during their fasting phase. And you can see on the right how meal times are narrowly distributed in the time-restricted groups. Whereas in the ad libitum fed mice, they ate mostly at night, but they also snacked kind of throughout the day. Now, as before, meal times seemed more randomly distributed across 24 hours when the mice ate a high-fat diet ad libitum, so whenever they wanted to. And what's important is that the amount of calories that they consumed over 24 hours is the same in all of these groups across the experiment. The only difference is in the timing of when they eat. And you can see even though they ate the same amount of high-fat diet over 24 hours, the time-restricted group was spared the massive weight gain. They're also spared the pathophysiological consequences of eating an ad libitum high-fat diet, like hepatic steatosis. And their livers look like the normal healthy livers of the chow-fed mice. Now, interestingly, locomotor activity patterns were also impacted by this meal schedule in both time-restricted feeding groups. And they even measured athletic performance here. This was assessed by their time spent on this accelerating rotor rod. So this data suggests that the time of feeding impacts locomotor activity levels and performance, but it's not clear exactly what's going on and how these are related. Now, to get some additional insight into relationships between feeding time and activity patterns, we used a model of progressive caloric restriction, which can be considered like an extreme version of this time-restricted feeding. So caloric restriction without malnutrition is known to extend lifespan and delay the onset of age-related disorders in a wide range of different species, including worms, mice, and even non-human primates. So for this experiment, we fed the mice an ad libitum chow diet, or we slowly reduced their caloric intake over 8 weeks to 60% of the ad libitum group. And you can see at the bottom the CR group, so the caloric restriction group, they lost lean mass and fat mass compared to the ad libitum fed group. So overall, they got quite a bit slimmer. And we used the DVC system to monitor locomotor activity throughout this experiment. I didn't try to figure out how to switch the default colors, so bear with me here. But here the green line reflects ad libitum fed mice, and the blue line is the caloric restriction mice. And this is the first week of the experiment, so everyone is eating roughly the same amount of food ad libitum. In fact, there's really no difference between these groups, and they're essentially the same at this point. So they wake up, they start eating their first meal during the first hours of lights off, and then right again before lights on towards the end of the dark phase. Now, when we start reducing the amount of food by 10%, you can see we immediately start to see changes in their activity patterns. So this increased activity is especially evident during the first hours of the dark phase, so when the mice are awake and anticipating the arrival of their meal. Now, we weighed their daily food intake of the ad libitum-fed group at the same time we fed the caloric restriction group, so both groups are disturbed the same way and at the same time every day. Now, as we get towards the end of the experiment, you can see this massive spike in activity during the hours immediately before feeding, followed by reduced activity towards the end of the dark phase. So at this point, the caloric restriction mice are eating 40% less than their ad libitum-fed counterparts, and they're very happy to see us when we feed them every night. They eat their entire meal in the very first hours upon arrival, whereas the ad libitum group still eats these two main meals, so one at the beginning and one at the end of the dark phase. So if we're to collect tissues from these two groups and compare them, we may in essence be comparing two very different nutritional states and two very different metabolic contexts, depending on when we make our comparisons. So of course, there may be no right or wrong way to select the time points for this tissue comparison, but it's extremely important that we take these relationships into account when we attempt to interpret any gene expression or metabolic data that comes out of these caloric restriction experiments. But when we examine the effect of caloric restriction on the cumulative activity, you can see we get only around 10% increased total activity by the end of our experiment, so after around 60 days. So any beneficial impact of locomotor activity on mice under caloric restriction is most likely due to the distribution of locomotor activity and consolidation into this very narrow window around feeding time, rather than this relatively modest increase in total activity levels. Now, we can visualize this progressive increase in food anticipatory activity if we look at the whole experiment as a heat map with consecutive days stacked one on top of another. So under caloric restriction, we start to increase activity during the final 2 hours prior to darkness. Towards the end of the experiment, this becomes very noticeable, turning yellow or orange and red even in the hours immediately before mealtime and lights off. And you can also see the acute effect on locomotor activity of the days when we took the mice out to measure body composition by echo MRI here, circled in red. So this suggests that if we do this intervention, this echo MRI measurement, we should wait a few days for these effects to normalize before we collect any tissues, as we may risk introducing confounding factors associated with this acute stress from handling the mice. Now, this data was also useful to identify an unscheduled cage change in the ad libitum fed group. Now, the online monitoring, again, allowed us to avoid introducing these confounders into our experiment. And the data suggested, again, that we should wait some additional days for this effect to normalize before proceeding with our tissue collection. And the final couple of slides, I want to briefly show a specific example of how passively monitoring circadian behavior was useful for our study design and for simplifying our workload. So in this case, we identified a circadian clock-controlled gene that codes for an important muscle receptor known to promote muscle growth at the cost of fat mass. And these effects can be induced pharmacologically by injecting a specific ligand, but the relative effects can vary and may take weeks before they're evident. So to validate whether oscillation of this gene is reflected at the protein level, we performed a radioligand binding assay here. And you can see we detected the highest protein content during the middle of the night and the lowest content in the middle of the day. So the protein data perfectly fits with our gene expression data, suggesting that the regulation of this receptor's 24-hour bioavailability may be an important clock-controlled process, and that the 24-hour differences in abundance may underlie the variation in the effects that are observed after pharmacologically activating this receptor. And we wanted to see whether we could harness this information and design a more targeted therapy. So for example, if we timed administration of the ligand to midnight when the receptor is most abundant, would we see a greater response than if we administer it at noon when it's 3 times reduced? And I have 2 small kids at home, so the idea of having to come in daily for 4 weeks straight to give treatments at night and at midnight and at noon was not appealing to me, nor was it appealing to my wife. So I decided to make a compromise. And I thought that if I shifted the lighting schedule of our mice by 6 hours, I could realign their circadian clock so that I could do my treatments at a single convenient time point, like say 6 p.m. every day. But their internal clocks would think it was either noon or midnight, depending on how I shifted them. So fortunately, our institute had purchased these LED light boxes that allow us to program the light schedule and intensity for each individual cage. So I could shift the light and wait for the mice to shift their clocks. So once I was sure they were shifted, then I could do my treatments and get good, reliable data and keep my family happy. So it seemed like a pretty simple solution, right? Well, actually, no. I found out the hard way that it takes longer to advance a clock than it does to delay a clock. And this is known for humans in that the duration of jet lag depends on the number of time zones crossed and the direction of travel. And re-entrainment is typically slower following eastward than it is westward travel. So on the left, you can see normal lighting schedule with activity segregated to the dark phase. And there's a spike in activity towards the bottom when I disturb the mice, again, to perform this echo MRI. Now, things are a little more messy with the data from the light-shifted mice in the middle and on the right. So the big red arrow at the top indicates the time when I started this shifted light schedule. And you can see in the blue line consecutive days slowly shifting right or left, depending on when the lights came on in the cage. Now, these little red circles are indicating my learning curve about when I should charge the batteries of my these LED boxes. So they should normally last a good couple weeks, but I wanted to first test the lights to see how long they can hold a charge and the daily percentage of the battery consumption between different numbers of days. So in any case, it's clear that recharging the battery disturbs the mice, and I should organize this event for the respective dark phases when the mice should normally be moving around in their cages anyway. So if you look at the dark blue line, you can see that the phase delay group, so the noon group, and this is the one mimicking east to west jet lag, seemed to adapt to the new lighting schedule within about 1 week or 6 days, whereas the phase advance group, so the midnight group, took around 2 weeks to shift. Now, to make sure things were relatively stable and consistent between the groups, I waited an additional week until after everyone was sufficiently shifted to begin my treatments. And I'm writing up these results as we speak, and hopefully you can see the results published in the near future. So I just want to quickly summarize some of the main take-home messages from my talk today. So first, I hope I've convinced you that time is very important, whether it's time of day, time of feeding, time of activity, the time it takes to adapt to a new circadian schedule, and even that there's often optimal treatment time to administer a therapy. So chronopharmacology is a relatively new field of study, but it has huge implications, since many FDA-approved drugs are known to directly or indirectly target components of the core clock machinery. Second, monitoring locomotor activity can provide important information about the relative circadian alignment of the mice, ensuring that the quality of the data collected is high and providing a solid foundation for study design and data interpretation. And finally, activity monitoring during an experiment, so online, can give important insight about potential problems and how to best avoid them. So I'm going to be sure to thank all my collaborators and thank you for your attention.
Excellent. Well, thank you, Ken, for the fantastic presentation. And with that, very pleased to introduce our second presenter, Dr. Joanna Moore.
Thanks, Liam. Thank you for inviting me to give this presentation. Today I'm going to talk about using home cage monitoring to determine the impact of timed mating on male mouse welfare. For this study, we used a strain of mice that produce naturally sterile hemizygous males called prim1s. If you're interested in what a prim1 mouse looks like, then this is an example of one. To identify a prim1 mouse, we don't need to genotype, as we can tell from the coat colour. The males and females are always agouti, and the extremities are bright pink because they express red fluorescent protein. Although only the males are sterile. If you're interested in learning more about this strain, please look at the paper on the results pane. All animal studies were ethically reviewed and carried out in accordance with the Animal Scientific Procedures Act 1986 and the GSK policy on the care, welfare and treatment of animals. The use of a naturally sterile male mouse is already a good refinement, as we no longer have to use surgery to create them. We further refined the model by always housing them with a companion female, as the males can be kept until they're 9 months old. And under the UK law and GSK's Code of Practice for Animals, we always house social animals in pairs or groups where possible. These males are used to induce pseudo-pregnancy in females within our animal facilities. The females tend to be swapped in the morning as part of the daily routine. And after 24 hours, the naive female is removed and the original companion is put back with the male. For the study I'm going to talk about, we left the naive female with the male for a couple of days so we could gather more data. We cannot be certain by visual assessment alone the impact of adding a naive female and removing the companion female will have on cage activity, and we hope that using home cage activity monitoring may help give us an objective answer. There are publications which have shown that high increases in cage activity are related to the disruptions caused by other potentially high activity generating interventions, such as cage changing and procedures. For example, Perno et al. Suggested that procedures involving handling or the transfer of animals disrupts their basic activity pattern and induces changes that appear to impact them over an extended period of time. And this could have an effect on the mouse welfare. So we use the DVC monitoring system to determine how much disruption is created to the activity pattern in the cage during the time mating procedure, when a companion female is swapped for a naive female, and compared it for when a companion female is briefly removed from the cage and replaced in the pair's home cage. Our overall aim for this study was to show whether cage activity significantly increases when a companion female is replaced with a naive female. To do this, we had two study objectives. One, to determine if the naive female causes a disruption in cage activity, and following on, how long it takes from the activity to return to a pre-swap activity level. First, I wanted to talk a little about working with big data, as this was one of the first studies we carried out using the DVC. We learned a lot about how to work with large amounts of data during these early investigatory studies. We realised we had to adapt the experiment to the DVC so that we were using it properly and would not get a false negative or false positive result, or a really unexpected answer to our research question, and maybe one that we thought was correct when it wasn't. One of the benefits of the DVC is the data can be downloaded to a readable Excel file, and it's relatively easy to select the timeframes of data you want to download. But with so much data generated, it's important to construct your scientific question prior to the start of the study. It's also worth noting that for the studies we used, we treated the cage as the experimental unit rather than the animal, because we couldn't tell the individual animals apart from the DVC data. DVC itself is a non-intrusive tracking system. You don't have to use microchips, there's no interference with the animals, and it sends data from the rack to a cloud storage via the master computer, which you can then download. The data is the animal activity index measured for all 12 electrodes per minute. In the picture on this slide, you can see the position of the 12 electromagnetic sensors, which continuously track and monitor the spontaneous activity of the mouse. In this example, I've tried to imitate the activity pattern of one male mouse in a cage. 0% means the animal is not moving at all over the observation period, in this instance 1 minute. And 100% means the animal was able to activate all of the electrodes for the entire minute of the observation. The 100% value is actually impossible to get, as it means all of the electrodes are activated for the entire minute by one animal. The animal movement is activating all the electrodes and changes the status of an electrode to active rather than inactive, and vice versa. So if the mouse becomes inactive, the electrode becomes inactive. The system is calculating the overall activity on the board and averaging the time period you are using. If you aggregate by an hour, it's 60 times 60 times 4 samples, or as we did in this study, by a minute is 60 times 4 samples. Therefore, the summary average activity is the average over 1 minute, which is the aggregation time we used. It is important to have a good understanding of the activity of the mice you're studying so that you can have a look at the activity, what activity they're doing across the day and night periods. This will then help you decide what activity period you want to study, which will give you the most accurate answer to your scientific question. After initially reviewing the mouse activity pattern for the animals we included in this study, we noticed that they had a very sporadic activity across the day and night periods. This could be related to social interactions within the pairs or because the unit is particularly busy in the mornings. This meant that data for each cage had a lot of variability over time and a lot of noise, so we selected 2-hour time intervals rather than a whole 24-hour period. The study ran over 10 days and the experimental unit was the cage. We included 20 cages of established, proven, sterile Prim1 males with their companion females and 10 naive CD1 weight-matched females. The mice were housed in GM500 individually ventilated cages on a digitally ventilated rack, the DVC from Techniplast. At the end of the study, the mice were all moved to a conventional IVC rack. Each of the cages contained our standard enrichment for animals, and they were housed under a light cycle of 14 hours light and 10 hours dark. The reason for this is because they were in the breeding unit. On the first day of the study, all of the cages were cage changed. This was to remove the confounding effect of cage changing. We know that this is a confounding factor because of a previous study where we could see the peak of the female swap was visible when we were cage changing. A note of the time of when the last cage was replaced on the rack was made, and we compared it to what time the DVC had recorded it. All of the activity was between 7 and 8 AM on the day of the swap. For the group 1, the cage was removed from the rack and the companion female replaced with a naive female. For group 2, the cage was removed from the rack and each female was removed from the cage for 5 seconds and returned to the home cage. Both of the interventions took between up to 10 seconds for each cage. And on the last day of the study, all of the naive females were replaced with the original companion female and the study ended. For the activity samples, we focused on the following 4 2-hour time intervals. 8 till 10 AM, because this was when the mice were very active due to the human occupation of the room. 10 to 12 AM, which was when the mice became less active. 12 to 2 PM, when the mice were at their most inactive during the day, and 8 to 10 PM, when the mice were at their most active. For each time interval, we calculated the change from pre-replacement and post-replacement activity for each cage to determine the activity increase or decrease on the day of female replacement, compared to the previous or subsequent days in the same 2-hour intervals. Initially, I wanted to show you the heat map. Group 1, where the females were swapped, is the heat map at the top, and Group 2, where the females were returned to the males, is the heat map at the bottom. You can see on this heat map, especially if you think back to Ken's heat map, where his mice had a very clear and consistent wake up and rest time period, how spiky the activity is for these animals, especially when they're at their rest period. The boxes indicated 1 is where the cage change occurred and 2 is where the swap occurred. And you can see that there's a much more intense period of activity as a result of cage changing compared to when we carried out the female swap intervention. But for the intervention, the swap there is a lot longer period of activity. The heat map is a 5-minute aggregation, as it was easier for me to understand the patterns of activity compared to when I tried the 1-minute aggregation. This graph shows the increase in activity for group 1, where females were swapped, compared to group 2, where females remained with the male during the 10 to midday time interval. This analysis focuses on the pre-female replacement. The reference on the outliers of this graph is that the letter refers to the column and the number refers to the row. Part of our statistical test was to determine whether there was any correlation between cage position, rack, and activity patterns. If we look at these outliers, we can see the most frequent one is A4. But when we tried to find a correlation between cage position, rack, and activity patterns, we could not find one. We then compared the activity for each cage on the day of female replacement to the average activity across the previous days for the same time interval. The pre-swap activity was generally similar between both groups, and there was a significant increase in activity in Group 1 compared to Group 2 during the 10 to midday time interval on Thursday compared to the previous days, and Group 2 on the same day, Group 1 being the group where the female was swapped, and Group 2 when the female was returned to the male. We conducted a t-test on the change from pre-female replacement measurement with each 2-hour time interval on the day of the swap being compared to the average activity of the previous days in the corresponding time interval. We can see that there was a significant increase for Group 1 when the female was swapped compared to Group 2. Interestingly, there was an unexpected response in the 8 to 10 p.m. 2-hour time interval. Here there is a trend towards a reduction in activity for Group 1 during the time when the mice are usually active. This analysis focuses on the post-female replacement. As previously indicated, Thursday was the time of the intervention. For this analysis, the 2-hour time interval activity on Thursday was compared with the average 2-hour time interval activity across the subsequent days of the study. As before, we looked at a correlation between cage position, rack, and activity pattern and could not find one. Although there was more variability in the activity during the post-swap time interval between 8 to 10 a.m., we can see that Group 1 cages start to return closer to pre-swap activity over a couple of days. Group 1 again is when the females were swapped, and Group 2 when the females were returned to the male. The t-test was carried out as before on the change from the day of female replacement and compared to the post-female replacement for each 2-hour time interval, comparing the average activity of the subsequent days in the corresponding time interval. We can see that there was a significant difference for Group 1 when the female was replaced compared to Group 2 where the female remained with the male. You can also see that an unexpected response at the 8 to 10 p.m. 2-hour time interval as previous showed the trend towards a reduction in activity for Group 1 during this time where the mice are usually active compared with the subsequent activity. In an earlier study, we again compared swapping a companion female with a new female with just taking the female out of the male cage and replacing her back with the male for a few seconds. But for that study, we did it at the same time as cage changing. We were interested to know if we could still see the impact of activity of the female replacement if we were cage changing, which, as I said before, already causes a huge peak in activity over a prolonged period. And in fact, we could still see this big peak in activity with the animals where the female was swapped compared to the animals where the female was put back in with the male, even when they were being cage changed, which suggests this is an added increase, an added potential welfare impact on the mice. The activity pattern of this mice was very varied, and it may have been because they were mixed sex pairs or because there was quite a lot of activity in the unit in the mornings. The increase in activity suggests there has been a disruption of the normal activity of the mice as a result of the female replacement. We can be quite confident that it is a result of the female replacement because we could observe the males chasing the female around the cage. We don't know how long this activity continued for, but it was likely they may have been scent marking, more chasing over the course of the active period. And the decrease in activity between 8 and 10 o'clock at night from group 1 compared to group 2 after the female replacement was very likely because of the intense activity during the light phase meant that the mice were fatigued during the dark phase. The reduction in activity during the night period, when we was expecting the mice to be most active, was something we wanted to try to address. And it could be that the cage activity will return to a pre-swap activity sooner if the female is replaced nearer the end of the working day, especially with these particular mice because they started to get more active as the working day drew to a close. And this was observed on the heat map. So home cage monitoring produces activity data we cannot obtain through observations alone, but it can actually help us to improve and refine some of our husbandry and scientific techniques. As with all this sort of work, it produces more questions and more plans than it does answers. And we really want to try to test if time mating in the late afternoon will decrease the disruption in cage activity. The thinking behind that is, although there'd still be a big peak in activity, it's during their very active period for these animals. We also want to see if the activity would increase if a naive female is added to a cage, but the companion left in the cage too. And if you're interested in what we found when we explored this research question, please join me at the Techniplus UK DVC webinar on November the 4th, where I might give you the answer. A further investigation would be whether the increase in activity was mirrored when the companion female was returned to the male on the same day. Unfortunately for us, in this particular instance, the companion female was returned to the male and put back onto an ordinary IVC rack because we didn't have much space on the DVC and we had a lot of studies to get done. We'd also like to see if we get a similar peak in activity if we introduce 2 unfamiliar female mice, and whether the activity peak would reduce quicker, just to see if it was because of the mixed sex pairing or if it's just something that unfamiliar animals will do. We wouldn't do this procedure with unfamiliar adult male mice because it's very likely to lead to fighting. It would also be interesting to see if the same conclusions were found under reverse lighting. All of our interventions are carried out when the animals are meant to be sleeping and during their rest period. And actually, you could also use the heat map to see the periods of activity, and if there's a repeat of that activity over a few days, then you may be able to do an intervention when they're naturally awake during the sleep period. And perhaps using reverse lighting would help. We did consider this at the start of the study, but due to the other work we were doing in the room, we weren't able to use reverse lighting. The use of ultrasound noise monitoring would be interesting to test to see if the vocalizations made by the mice during the peak activity period were a sign of a positive experience. In a study with rats, it was found to be a positive vocalization, so it would be interesting to see if it would be the same with mice. And video data would enable us to be confident about which mouse was carrying out which activity and who was causing the activity and exactly what they were doing to create the peaks in activity over a longer period of time. But using video data alone, we would have to view the full video. But if we used it in conjunction with the DVC, we'd only need to check the video when there is a peak of activity. And this would be the same for ultrasound monitoring, as the DVC can be linked to other electronic devices. So home cage activity monitoring gives us a unique insight in terms of how the work we do can impact the welfare of animals, thus giving us an opportunity to refine our husbandry processes, scientific studies, and care to meet their future needs. And I'd really like to thank everybody that was involved in this project, especially Guido, Fabio, and Scott, who gave us really great support and help in learning and understanding how to work with the DVC and the data it generates. Thank you all for listening.
Thanks so much, Joe, for the excellent presentation. Okay, so let's jump right in. First question today is, how does home cage monitoring change the way that you perform research? Or I guess more specifically for the DVC, has the DVC changed your ability to conduct your studies? Maybe Joe, I'll direct this question to you first.
I think it's changed it completely, really, because we wouldn't even have been able to think of a way of looking at the impact of timed mating on animals or any impact on the activity of the animals without something like the DVC doing the home cage monitoring, and to be able to see the differences in activity when there's no human intervention and during their awake time when they're actually most active is really, really useful for our research, and going forward would be a real benefit for research.
Excellent. Thanks so much, Joe. And Ken, anything to add to that? What's your experience with this?
I mean, we've been using activity monitoring in different ways, more invasive than others sometimes. And this was a good, let's say, compromise in terms of being able to measure simultaneously many mice, so big cohorts. I mean, we're doing metabolic research, so there's a lot of variability in terms of when mice are eating, obviously, and how often they can become disturbed, as you saw today. So this is a way to really increase our numbers so we could reach and get some kind of physiological insight into, even though there is some variability between animals. So I think overall it's just helped us to monitor bigger cohorts and get better data.
Excellent, great answer. And I guess that covers the next question a little bit, but maybe we could talk about that a little bit more. But in your experience, what are the main benefits of the DVC, and what are the trade-offs? What are some of the challenges associated with this research? Maybe Ken, I'll direct this one to you first.
Yeah, so we have these metabolic cages, so we can measure oxygen consumption, food consumption. We have some running wheels as well. The DVC is not measuring all of that, but it does let us measure activity, and it does let us monitor at least these behavioral patterns. The other methods, I think we're kind of stuck with using small cohorts because we don't have unlimited metabolic cages. I mean, we have quite a few, I think, but I mean, not the same as a whole DVC rack, let's say. So this is definitely one of the big trade-offs for moving to the DVC, is just more numbers and getting nice activity data.
Excellent, thanks. And Joe, what's your experience? For your research, what do you think are the main benefits and major trade-offs?
I think the main benefits are being able to see the animals 24 hours, and seeing them the activity live as well, and being able to produce heat maps and all of the data that the DVC can download. But equally, you can look at specific periods of time, and you can put lots of different animal, you can put cages on the rack and observe all of them. It gives you a real 24-hour insight into the activity, and it will send an alert if there's any problems in terms of a sudden reduction in activity, which is also a good benefit.
Excellent. Thanks, Joe. Next question here. We've received actually several questions about data collection and analysis. For example, how easy do you find data collection and analysis using the DVC? Would it be easy, for example, for an inexperienced technician or a grad student to pick up? And Joe, I'll let you lead this question.
I think it would depend on their background experience. They would need a certain amount of understanding of statistical packages, because while the DVC data is the interface of the DVC analytics is very user friendly, in my opinion, and when I've been using it, and it will give you some answers, but it doesn't give you the full statistical package that you need. So ideally, you can download it quite easily, and you can choose the time frames that you want and what animals you want to get, what cages you want to observe. Download it into an Excel file. But I was very fortunate that I had a great statistics team to help me, and I think you really need some understanding of statistics. And one of our MSc students is actually he's a statistics MSc student, but he's doing his MSc with the data from the DVC for this study actually that I'm talking about.
Excellent. Thanks, Joe. And Ken, what's your experience like with data collection and analysis? Do you find it fairly simple, or do you think it would be easy to pick up?
Yeah, it's pretty straightforward to do the online monitoring for sure. I can figure it out. I'm not too tech savvy, but I think my problem is I'm trying to run many studies simultaneously, filling up the rack with different cohorts at the same time. This is another feature, let's say, that is a good thing and a bad thing in the sense that you accumulate data quite fast. And so it does take some time, like in any study, to sit and parse the data and organize the cages. This is pretty straightforward with the user interface, but I'm a little bit lazy, obviously. So I tend to accumulate data and then wait till then I find time and motivation to download it. So I guess this is a good thing and a bad thing about having all the data on the cloud all the time.
Excellent. And Ken, actually, a question came in during your presentation. Can you just comment on how the locomotion index is extracted from the locomotor DVC data?
Yeah, so the data itself is average of all of the groups, or all of the mice in the group that I showed today. You can do this individually per cage. You can bin it in different ways. You can bin it according to minutes or hours, or only the light phase or only the dark phase. This you have to set up on the user interface itself. But then there's different options also for the visualization itself. Yeah, to group all of the let's say my experimental groups together, this is pretty straightforward. And I think the visualization is the average activity of all of the individuals within that time frame that I set.
Excellent. Thanks, Ken. And here's a question that we've got a couple times. And Joe, I'll ask you to lead this one, but how many mice can be assessed at a time per cage?
Well, we have, I would say up to 4, up to 4, but it works best if you have the same number of animals per cage, but I would say 3 to 4.
Excellent. Thanks, Joe. And Ken, what have you found for this? What's the maximum number of animals you've tried in one of these cages?
So I'm trying to look for outliers because, I mean, mice are like people. Some of them like to move more than others. So to identify these, we try to keep it to a minimum. So we do single house the male mice for the duration of the experiment, but this is only like a week or so. And I think the data is only the one mouse in that case. We have a colleague here that is monitoring, like Joanna, multiple mice in the cage, and because of the location within the grid, you can identify individual data points, I think. But I haven't been doing this.
Excellent. Thanks, Ken. And I think in the interest of time, we'll make this next question the last one. The question being, have you used these methods for investigating drug effects? If so, how did you do this and what did you find? Maybe Joe, I'll ask you to lead this one.
Unfortunately, we were going to use this for exactly that type of study, but due to the COVID, we had to stop our DVC work for a while, so not yet, but hopefully we will be. It certainly has been done by other people. If you go to the DVC webpage, there's some papers you could look at.
Excellent, thanks. And Ken, do you have any experience with using the DVC to investigate drug effects?
I would say, unfortunately, yes, in the sense that we do have to administer some drugs sometimes, and you have to be quick. I mean, it's a normal IVC cage system, so you have to be as quick as you can open and inject the mouse and put it back in there. But yeah, you can obviously compare the same individuals longitudinally before, during, and after the treatment. So I think, yeah, it's actually very well suited for these types of studies.
Excellent. Well, thanks so much, Ken and Joe, for all your fantastic insights today, both in your presentations as well as the Q&A session here.
Thanks for having us.
Yeah, thank you.
Yeah, and a big thank you to everybody attending today for joining us. And last but certainly not least, a very big thank you to Technoplast for sponsoring the event. Now, in closing, thank you again for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon. Have a wonderful day, everyone.